Hello, am I audible? Okay. So, hi, I'm Shrija Ghosh. I'm a PMRF from IIT Kanpur, and I'm taking the problem solving sessions for the NPTEL course Environmental Chemistry and Microbiology. The course instructors are Professor Anjali Pal and Professor Sudha Goel from IIT Kharagpur. And today is the 11th problem solving session. And today we'll discuss the questions from different microbial diseases, different pathogens, and the disease they cause. And we'll start with the metabolic diversity of the microorganisms. So we'll discuss a few photosynthetic pathways and questions related to that. Okay. So I'll start with the questions. Just let me know if you have any doubt. You can either write or you can unmute your mic and ask. So question number 1. Ringworm is a disease caused by option A fungus, option B bacteria, option C tapeworm, option D roundworm. So anyone wants to answer? If you want to answer the question. Ringworm is a disease caused by So ringworm is a disease caused by fungus. So the correct answer for this question is option A fungus. So ringworm, also known as dermatophytosis or dermatophyte infection, is a fungal infection of skin. The term derma is generally associated with skin. So dermatophytosis or dermatophyte. The scientific name for this disease is Tinea corporis. And ringworm infection can affect both humans and animals. And the infection initially appears as red patches on the affected area of skin, and later may spread to other parts of the body. It may affect the scalp, feet, nails, groin, beard, or other areas. So mostly what happens on our skin, these fungus, it causes infection on the skin surface, and you can see a round shaped patch, red colored patch forms during this infection, and that patch like later after you put medicine over it, it will slowly die, the pathogen will die, and then the inflammation that has happened on the skin will slowly dry off, and then it will go away from the skin. Okay. So moving to the next question. If you have any doubt, you can either unmute your mic and ask, or you can write in the chat box. So the next question is, teardrops are rich in option A amylase, option B phosphate, option C lysozyme, and option D protease. So teardrops, they have one particular component. Yes, Amadeep has answered option C, lysozyme. So that is the correct answer. So teardrops are actually rich in lysozymes. Can you tell what other secretion of our body is rich in lysozyme? Can anybody tell where else we can see lysozyme in our body? So our saliva is also rich in lysozyme. So lysozyme, it's an enzyme found in the secretions, that is tear of the lacrimal glands. Lacrimal glands are the glands that in which these tears are produced. So it's a type of secretion of our body, right? So lacrimal glands of animals, and in nasal mucus, gastric secretions and egg white. So apart from the lacrimal gland secretion, it is also found in nasal mucus, gastric secretions and egg white. Discovered in 1921 by Sir Alexander Fleming, lysozyme catalyzes the breakdown of certain carbohydrates found in the cell walls of certain bacteria. So presence of lysozyme is kind of a protective measure which is naturally found in our body. So since our tears contain these lysozymes, and other than tears, our other body secretions also contain these lysozymes, it's kind of a non-specific protective barrier that is provided by our body. So lysozyme can break down the cell wall of certain bacteria, and hence it can protect us against some pathogenic bacteria from entering our body. It can prevent them to enter our body. Okay. So moving to the next question, question number 3. What is the importance of the epidermis regarding human immunity to microbes? What is the importance of the epidermis regarding human immunity to microbes? Option A, it is the first layer of skin which helps in defending the human body from microbes. Option B, epidermis keeps the body temperature sufficient to kill the microbes. Option C, there is no specific role for epidermis to prevent in preventing diseases. And option D is, it is the last layer of skin which helps in defending microbes from reaching the human body. So the correct answer, yes, Chetna has answered. The correct answer is option number A. It is the first layer of the skin which helps in defending human body from microbes. So human body has certain protective barriers. So skin is the first barrier that protects our internal organs and tissues from different microorganisms and different pathogens present in the environment. Okay, so this is diagram of the skin. So the outer layer is called epidermis, and the inner layer is so the layers sorry. So the outer layer is called the epidermis, and the inner layer is dermis. Human skin has 3 main layers. The epidermis is the outermost layer. 2 other layers of the skin are dermis and hypodermis. Epidermis is the thinnest layer of the skin, but it's responsible for protecting a human being from outside world, including pathogenic microbes. So basically our skin acts like a barrier for any pathogen to enter. So that's why if you get a cut in the skin, it is advised to put some antiseptic, and so it is advised to put some antiseptic so that you can prevent any microbes. Since the epidermis is broken, there are chance of other pathogenic microbes entering your body through that broken skin, and it can cause infection. So to prevent such infections, we generally use antiseptics on the wounds. So that is the reason. So skin is the most important protective barrier for your body. Okay. So moving to the next question, question number 4. What causes bubonic plague? Option A, Streptococcus mutans. Option B, Porphyromonas species. Option C, Prevotella intermedia, and option D, Yersinia pestis. Which of the following microorganisms causes bubonic plague? Any idea? Anyone wants to answer? Yes, the correct answer is option number D, Yersinia pestis. So, bubonic plague is one of the 3 types of plague caused by plague bacterium Yersinia pestis. So, it is also known as plague bacterium. It produces a toxin called plague toxin, which actually causes this plague, this bubonic plague. So, it is an infection spread mostly to humans by infected fleas that travel on rodents. So, there is a flea called an oriental rat flea, Xenosylla chiopus. It basically this flea is infected by the bacterium Yersinia pestis. So, this flea catches this bacteria, and now this flea acts as the vector and carries this diseases. This flea carries the disease to different rodents, basically rats and squirrels, like that. And then from those rodents, if a person gets rat bite or something, that person can get basically this bacteria Yersinia pestis, it will get transferred from the flea to rodent and then to humans. Okay. So, this Yersinia pestis, it is found in the gut of this flea, and it appears as a dark mass in the gut. So, in the figure you can see here, in the gut of the flea, the foregut of the flea is blocked by Yersinia pestis biofilm. When the flea attempts to feed on an uninfected host, Yersinia pestis from the foregut is regurgitated to the wound causing infection. So, when let's say this flea bites a rodent, the bacteria, the plague bacterium from the gut of this flea, it enters the rodent's body through the bite infection that the flea caused. Okay, and from the rodent then it passes to the humans when the rodent bites the human. Okay, so this is clear. In this topic there are a lot of things that you have to remember. You like the name of the bacteria and the disease caused by that bacteria, those things you have to learn and remember. So, the next question, moving to the next question, question number 5. Proteus species affect dash tissues and causes dash disease. So, which tissue is affected by Proteus species and what disease is caused by this in that particular tissue? So, option A is for the tissue, it's throat epithelium, and the disease caused is pyelonephritis. Option B is the tissue is small intestine epithelium and the disease is cholera. Option C is the tissue is kidney medulla and the disease is pyelonephritis. And option D is again tissue is kidney medulla and the disease is spontaneous abortion in cattle. Yes, Chetna has answered. The correct answer is option C. So, Proteus species affect kidney medulla and causes pyelonephritis disease. Okay, so what is pyelonephritis? It's a type of kidney infection. So, this is a healthy normal kidney. So, these parts are the kidney medulla. And you can see this is the affected kidney after pyelonephritis, and you can see the kidney medulla is totally infected. You can see the scar and pus formation in the kidney medulla. So, it is totally inflamed. Okay, so kidney infection, that is pyelonephritis, is a type of urinary tract infection that generally begins in the urethra or bladder and travels to one or both of the kidneys. Okay, so this Proteus species, it basically starts the infection from the urethra or the bladder, urinary bladder, and then it travels all the way up to the kidney. Okay, and spreads the infection there. So once it reaches the kidney gets inflamed, and you can see the inflammation majorly occurs in the medulla region of the kidney. Okay, this is clear, I hope. So the next question is question number 6. Which among the following is not a host to host epidemic? Is not a host to host epidemic. Okay, so you have to pay attention. So option A is H1N1, option B is hepatitis, option C is influenza, and option D is COVID-19. So which of the following disease is not a host to host epidemic? So I think we all know the spread of COVID-19, so that is surely a host to host epidemic. I think you would agree with that. Other than that, the three options are influenza, hepatitis, and H1N1. So influenza, again, it's the virus that causes common cold, so it's also a host to host epidemic disease. Yes, Chetna has answered option number B, so hepatitis. So H1N1, again, it's swine flu. You remember swine flu was caused by H1N1 virus, so that was also a host to host spread disease. So hepatitis is the correct answer. It is not a host to host epidemic. So hepatitis A is an inflammation of the liver caused by hepatitis A virus. It is also a viral disease. The virus is primarily spread when an uninfected and unvaccinated person ingests food or water that is contaminated with the feces of an infected person. So it doesn't spread from host to host, it spreads from the contaminated food or water, and the contamination is basically fecal contamination of an infected person. Okay, so the disease is closely associated with the unsafe water and or food, inadequate sanitation, and poor personal hygiene. Okay, so this is how hepatitis A spreads. So all the other diseases, like COVID-19, or in case of common cold, any influenza, flu, or in case of swine flu, all these flu can spread from one host to another through physical contact, right? If I sneeze and my droplets, if I am already infected, my sneeze droplets can get transmitted to the next person in front of me, and the disease can spread from one host to another easily. Okay, but in case of hepatitis, it won't happen like that. In case of hepatitis, generally, if you are having some street food which is uncovered, and then some kind of flies are sitting on it, and the flies are already contaminated, it has some fecal contamination of an infected hepatitis patient. So basically, if the fly has this virus in contact and then it sits on some food, so then that's how the food gets contaminated with hepatitis A, and if you are eating that food, it can cause infection in your liver as well. So that is how, so it is not directly spreading from one host to another. I hope that is clear. So next question is question number 7. Identify the vector of Lyme disease. Option A, Aedes mosquitoes. Option B, Anopheles mosquitoes. Option C, black-legged ticks. And option D, cattles. So identify the vector of Lyme disease. Which of the following is a vector of Lyme disease? Any answer? Aedes mosquitoes, Anopheles mosquitoes, black-legged ticks and cattles. So the correct answer for this question is option C, black-legged ticks. So can you tell me what diseases are spread by Aedes mosquitoes and Anopheles mosquitoes? Anybody? So Aedes mainly spreads dengue and chikungunya virus. So Aedes is the vector for those two viruses and Anopheles is the vector for malaria parasite. So these two mosquitoes are mainly associated with all these diseases. You must have heard that malaria, dengue, these are caused by mosquitoes. So these are two different species of mosquitoes. Aedes mosquitoes carry the dengue virus and the chikungunya virus, whereas Anopheles mosquitoes carry the malarial parasite, okay, plasmodium, that causes malaria. So black-legged ticks carry the so black-legged ticks are the vector for this Lyme disease. So Lyme disease is the most common vector-borne disease in the United States. Lyme disease is caused by the bacterium Borrelia burgdorferi. So Lyme disease is caused by the bacterium Borrelia burgdorferi and rarely Borrelia mayoni. It is transmitted to humans through the bite of the infected black-legged ticks. Most common sign of infection is an expanding red rash known as erythema migrans that appears at the skin of the tick bite about a week after it occurred. So here's a photo of the black-legged tick. So these are small insects and they can, if they are infected by the by this bacterium Borrelia burgdorferi or Borrelia mayoni, they can actually transfer that bacterium when they are biting the humans. They can transfer the bacterium from their body to the human beings and thus causing the Lyme disease in the human. So how in case of malaria, you know, when the infected mosquito that has the plasmodium parasite, it bites the human beings, it leaves the plasmodium eggs in the human blood, and then the plasmodium goes and affects the RBCs of the human and causes malarial disease. So similarly in this case, black-legged ticks actually bite the human, and if they are carrying this Borrelia species, so they will transfer the Borrelia species from their body to the human beings, and then same how malaria develops, and similarly Lyme disease can develop in such human beings that are bitten by black-legged ticks infected by the Borrelia bacterium. Okay, I hope that's clear. You can ask if you have any doubt. So the next question is question number 8. Identify the non-zoonotic diseases from the following. Option A, anthrax. Option B, rabies. Option C, salmonellosis. And option D, malaria. Which of the following is non-zoonotic disease? Do you know what a zoonotic disease is? Yeah, Chetna has answered option D, malaria. Malaria is a non-zoonotic disease. What do you mean by zoonotic disease? Amardeep has answered option A, anthrax. Can any one of you Tell me what a zoonotic disease is. Yes, it is originated from animals. Yes. So, correct answer is anthrax. Right now, I told you that malaria is transferred from, like it is transmitted through a vector, right? So, any vector borne disease won't be considered as non-zoonotic disease. It will also considered as zoonotic disease. Okay. So, zoonotic diseases are the disease that are transmitted from animals to humans. So, anthrax, it is a serious infection caused by gram-positive rod-shaped bacteria called Bacillus anthracis. So, Bacillus anthracis is the causative agent for anthrax. It actually produces a toxin. So, toxins are some harmful compounds, you can say, that are produced by these bacteria, which when transmitted to the host, it can actually develop a disease. So, it's a poisonous substance, you can say. Okay, it's a toxic substance that is produced by a bacteria. So, Bacillus anthracis also produces a toxin which causes this anthrax. So, anthrax can be found naturally in soil and commonly affects domestic and wild animals around the world. Inhalation of anthrax can occur when a person inhales the spores that are in the air during the industrial processing of contaminated materials such as wool, hides or hair. So, these Bacillus anthracis actually they produce spores, okay, and since they can produce spores, their spores can be dispersed in the air in aerosolized form, okay, in forms of aerosol. The spores can be suspended in the air around, and these spores are very resistant structures. Actually, they form these endospores, and these spores are very resistant. They don't get destroyed very easily in the environmental conditions, like in the harsh environmental conditions also these spores do not get destroyed easily. And if a person inhales these spores, the person can actually, so once a person inhales these spores can germinate inside the host body, and then it can cause the infection. Once the spores germinate, it will make the live bacterium again, Bacillus anthracis, and it can produce the toxin, anthrax toxin, that can cause anthrax in the host body. Okay. So, the other options were rabies, salmonellosis and malaria. So, rabies, you must have heard, generally people say if a dog bites it, you can get rabies. So, not just dogs, there are many rabid animals, like rabies is a virus actually, and a lot of animals are host of these virus. So, rabies virus is transmitted through direct contact, such as through broken skin or mucous membrane in the eye, nose or mouth, with saliva or brain or the nervous system tissue from an infected animal. So, let's say a dog is infected by this rabies virus, or a rabbit is infected by this rabies virus, and then when the dog bites the human, or even through his saliva, like through the dog's saliva, this virus is present in the saliva of these infected animals. So, if a dog bites, now my skin is broken. I have told you during the first or the second question that our skin acts as a protective barrier, but if there is a cut in it, now the skin is broken. So, any infection causing agent, any pathogen or any microorganism, it can now enter through our skin inside our body, right? So, when a dog is biting a human being, so because of the bite there is a cut in the skin, and now the skin is open, and if the dog's saliva has these rabies virus, So while it is biting the human, the virus can enter the skin, enter the body through the bite mark in the skin and cause the infection. And this rabies virus generally affects the nervous system of our body. So if you would have noticed, some of the street dogs are very aggressive in nature. So those dogs might have this rabies virus that makes them so aggressive, because it is affecting their brain, their nervous system. It tends to make it tends to give such effects to the infected animal. Okay. The next disease in the options was salmonellosis. So salmonellosis, again, it is caused by a bacteria called salmonella. So salmonella are a group of bacteria that can cause gastrointestinal illness and fever called salmonellosis. Salmonella can spread from animals to people. People who have direct contact with certain animals, including poultry and reptiles, can spread the bacteria from animals to food if they do not practice proper hand washing hygiene before handling food. So salmonella is also spread in the similar way rabies is spread actually. So it is generally present in these farm animals, poultry and all. So in those animals, this bacterium is present, and if you are handling such animals in a farm, and after that if you are not washing your hands properly, your hands or something can carry these bacteria, since you are working with those animals. And if you are not washing your hands properly, if you are not maintaining your hygiene, now that those bacteria from your hand can enter your body when you are eating with hand, or like can enter in your food, and from food it can enter in your body. Okay. So salmonellosis is a infectious disease caused by salmonella bacteria. And the last was malaria. I've already told you, malaria is a mosquito borne infectious disease. Anopheles is the female Anopheles mosquito are the vectors actually that affect the humans and other animals. Malaria causes symptoms that typically include fever, tiredness, vomiting and headaches. In severe cases, it can cause yellow skin, seizures, coma or death. Okay. So I hope all these diseases are clear, like what are the causative agent and how does it spread. If you have any doubt, you can ask. Okay, so the next question is question number 9. Sandflies spread the deadly disease which is known in India as Kala Azar. Which microbes cause this disease? So Kala Azar is a disease and the vector for the disease is sand flies. But what is the microbe that causes the disease? So you have to understand the difference between the vector and the microorganism causing the disease. Vector is something which is just carrying that microorganism, that pathogen, and transferring it to the other host. Okay, but the microorganisms that are causing the disease, they are the actual causative agent, they are the pathogens. So in case of malaria, mosquito is just the vector. The main culprit is Plasmodium falciparum or Plasmodium species, the parasite. Okay. So for this question, sandfly spread the deadly disease Kala Azar. Which microbe caused this disease? So Amardeep has answered option number A, right? So Leishmania protozoan So the correct answer is Leishmania protozoan. They cause this disease Kala Azar. So visceral leishmaniasis, also known as Kala Azar, is fatal if left untreated in over 95% of the cases. It is characterized by irregular bouts of fever, weight loss, enlargement of the spleen and liver, and anemia. Most cases occur in Brazil, East Africa, and India. Leishmaniasis is caused by protozoa parasite from over 20 Leishmania species. Over 90 sandfly species are known to transmit Leishmania parasites. So, it's a parasitic, it's a protozoan disease. So, Leishmania is a protozoan, and it's a parasite. Parasite meaning it cannot live on its own. It basically feeds on the host for its own survival, and in turn causes infection in the host. Okay, and the disease is characterized by irregular bouts of fever, weight loss, enlargement of spleen and liver, and anemia. These are the symptoms of the disease. Okay, just a second. Yeah. So this is clear. Leishmania protozoan causes the disease called Kala Azar or Leishmaniasis. Okay. The next question is question number 10. Amoebiasis is a waterborne disease. Which part of the human body is most affected by this parasitic disease? Option A, eyes, option B, ears, option C, urinary tract, and option D, colon. So, Chetna has answered option D, colon. So, the correct answer is option D, colon. So, amoebiasis, it's a waterborne disease, and it affects the colon of the human body. So, it is a parasitic infection of the colon or intestines caused by the protozoan Entamoeba histolytica or E. histolytica. So, it is again a protozoan disease. So, this protozoa Entamoeba histolytica, it enters the human body and causes the infection in the intestine of the humans. Symptoms for amoebiasis includes loose stool, abdominal cramping, and stomach pain. However, most people with amoebiasis won't experience significant symptoms. In some cases, it invades the colon wall, causing colitis, acute dysentery, and long-term chronic diarrhea. The infection can also spread through the bloodstream to the liver. So basically, this Entamoeba histolytica, it enters in the human body through contaminated food items and contaminated water, and once it enters the human body, it infects the intestines of the humans, causing loose stools, diarrhea like symptoms basically. But most of the times it gets cured by normal medication. Sometimes the infection, if not treated, sometimes the infection stays there, and then it can lead to more chronic diarrhea, or it can invade the colon wall, causing colitis or ulcer kind of situations. Okay, and it can spread to the liver also. From the intestine, it can reach the liver also if not treated at all. Okay. So the next question is question number 11. The relative ability of a parasite to cause disease is called dash. The relative ability of a parasite to cause disease is called dash. Option A is vector, option B is virulence, option C is virality, and option D is viral ability. So yes, the correct answer is option B, virulence. So virulence is a pathogen's or microorganism's ability to cause damage to a host. So virulence is basically the factor by which you can tell whether a microorganism or a pathogen is dangerous or not. So if you remember, during COVID pandemic, people were talking about different variants of the virus. Initially there was some Omicron, then Delta variant. So different variants of this virus were having different degree of virulence. So you remember when the Delta variant was causing the infection was more severe, and the patients were more in a more severe conditions, right? Whereas in case of Omicron variant, the cases were less severe. So you can say the virulence factor of that Delta variant of COVID-19 was higher than the virulence factor of the Omicron variant. So this is how this virulence is used to determine how severe the infection caused by that pathogen will be. If it is highly virulent, it can cause more damage to the host. Okay, I hope that's clear. Next question is question number 12. Microbe human interaction can be neutral, mutually beneficial, or pathogenic. Okay, in the following list of microbes, identify the mutually beneficial microbe out of the pathogenic ones. Option A, E. coli, Escherichia coli. Option B, Mycobacterium tuberculosis. Option C, Clostridium botulinum. And option D is Staphylococcus aureus. So there are three types of interaction mentioned in this question: neutral, mutually beneficial, or pathogenic. So we have to tell which of the following bacteria is mutually beneficial out of the pathogenic ones. Yes, Amardeep has answered option number A, Escherichia coli. So the correct answer is option A, E. coli or Escherichia coli. It's a type of bacteria that normally lives in the intestine of all warm-blooded animals. So it's a gut bacteria actually. It's present in our gut. Except E. coli, all other microbes given in the options are pathogens. So if you see Mycobacterium tuberculosis, it's in the name only, it causes tuberculosis, and it's a very dangerous bacterium. There is still no cure for tuberculosis, 100% cure, because there has been multiple cases of antibiotic resistance for these Mycobacterium tuberculosis bacteria. So the problem with curing tuberculosis is actually this bacteria, it enters deep inside the lungs, and any medicine that are given to treat this to treat the patient of tuberculosis, those medicines can't reach the bacterium and kill it. The second problem with this bacterium is it can stay in a latent phase. So if you ever go to some country where tuberculosis is not very prevalent, they will ask you to get a latent TB test, and most of the Indians, if you will do the test on them, they will have this latent TB, because in India TB has been there and it is still not eradicated from our country. So most of our body actually contains that latent TB, but it's not dangerous till it's latent. The problem is that latent TB can anytime become virulent and cause the infection. And because of antibiotic resistance, there is still no particular antibiotic that can completely destroy this bacteria. Third is Clostridium botulinum. So it also produces a toxin called botulinum toxin. So it's also a disease causing pathogen. And the last is Staphylococcus aureus. It also causes infections inside the mouth. Okay, so oral infections are caused by Staphylococcus aureus. So E. coli is the only or microorganism among the following options that is not pathogenic. It is already present in our gut. So the next question is question number 13. Organisms that can make their own energy using light and carbon dioxide via the process of photosynthesis are: option A, photoautotrophs; option B, chemoautotrophs; option C, photoheterotrophs; and option D, chemoheterotrophs. So I have told you this in I think 8th or 9th session also. What is the difference between autotrophs and heterotrophs, and then photoautotrophs, photoheterotrophs, chemoautotrophs and chemoheterotrophs. So organisms that can make their own energy using light and carbon dioxide via the process of photosynthesis, they are called, what will be the answer for this? Yes, option A, photoautotrophs. So the correct answer is photoautotrophs. These are the organisms that can make their own energy using light and carbon dioxide via the process of photosynthesis. So this is the diagram that I have shown in previous session also. So all organisms can be divided into two categories based on the carbon source they are feeding on. So if they are using CO2 as their primary carbon source, they are autotrophs. If they are using some other organic carbon source, they are heterotrophs. Okay. Now among the autotrophs, now based on the among the autotrophs and heterotrophs, now based on the type of energy they are using to break down to use this carbon source. So basically, if they are using organic carbon source, the type of energy they are using to eat that carbon and then produce ATP and metabolize that carbon source. Basically, if they are using light energy, the term photo is used as the prefix. If they are using chemical energy, the term chemo is used as the prefix. Okay. So among the autotrophs, there will be two categories, photoautotrophs and chemoautotrophs. Among the heterotrophs, again there will be two categories, photoheterotrophs and chemoheterotrophs. So based on the light energy, based on the type of energy, this division is made. After this, the photoautotrophs can be divided into two categories again. If they are using H2O to reduce the CO2 or the carbon source, you can say, since they are autotrophs, they are using CO2 as their carbon source. So if they are using H2O to reduce that CO2, they are oxygenic photosynthetic organisms. So they are growing in presence of oxygen. Okay. And if they are not using H2O, then they can use some other form of they can use some other form that can reduce the CO2. So H2O, in place of H2O, most organisms use H2S. So the organisms that cannot grow in oxygenic conditions, in presence of oxygen, they are called anoxygenic photosynthetic bacteria, like green and purple bacteria. They will use H2S as the agent to reduce CO2. Then if they are using chemical energy, then they will be called chemoautotrophs. So they can use again hydrogen, iron, sulfur, nitrogen and carbon monoxide to oxidize and in turn reduce the CO2. In case of heterotrophs, again you have two categories, photoheterotrophs and chemoheterotrophs. So in photoheterotrophs you have green and purple non-sulfur bacteria. So in case of photoautotrophs, you saw if it was not using H2O, if it was anoxygenic, that means in absence of oxygen, it was green and purple sulfur bacteria basically, because they are using H2S in place of H2O. Now in heterotrophs, these are green and purple non-sulfur bacteria, okay, because they are using the light source and organic carbon as the carbon source, okay. Among the chemoheterotrophs, now they are using the chemical energy and using the organic carbon as the carbon source. So chemoheterotrophs, if the terminal electron acceptor is oxygen, then it is the usual aerobic respiration, which we have studied in the previous sessions. So you have studied the electron transport chain and how ATP is being made, the whole oxidative phosphorylation. So most of the animals, fungi, protozoa, bacteria, they follow this aerobic respiration, whereas most of the plants and algae and some bacteria, they follow oxygenic photosynthesis. So this is the difference between the two, okay. Both are in presence of oxygen, but one is using CO2 as the carbon source, one, so basically they can produce their own organic matter, and one is using the organic matter as the carbon source and metabolizing it into CO2 in presence of oxygen, that is aerobic respiration. If the terminal electron acceptor is not oxygen, then you have two cases. It can either be some organic compound that for example some fermentative bacteria like lactobacillus, or it can be some inorganic compound. In that case, anaerobic respiration takes place. An example is Clostridium species, it's a bacterium, okay. So you understand. So the question was organism that can make their own energy using light and carbon dioxide. So you can see, using carbon dioxide as the carbon source and light as the energy source, these organisms can produce their own energy. Own energy means they can produce their own ATP, okay. Whenever we are using this term energy, we mean at the end the goal of any living organism or living being, whether it's plants, animals or microorganisms, finally the goal is to survive, and survival is possible only in presence of ATP, okay. So photoautotrophs are the ones that can use CO2 and light energy to produce their own energy, okay. Next question is question number 14th. Sorry. Question number 14th. Organisms that are able to synthesize their own organic molecules from the fixation of carbon dioxide and are able to produce their own source of food or energy are. Now I have explained the whole table, so you should be able to answer this. Organisms that are able to synthesize their own organic molecules. So they are synthesizing the organic molecules from fixation of carbon dioxide. So they are using carbon dioxide to synthesize their own organic molecule. That means they are autotrophs, right? And are able to produce their own source of food or energy are. So the answer would be Option B, chemoautotrophs. Because they are using carbon dioxide, they are autotrophs. They are using chemical energy because they are synthesizing organic molecules. So they are chemoautotrophs. Okay. So chemoautotrophs are the organisms that obtain their energy from chemical reaction, that is chemotrophs. But their source of carbon is most oxidized form of carbon, that is carbon dioxide. Okay. So the answer is chemoautotrophs. I hope this is clear. This is very important. You should know the difference between the autotrophs and the heterotrophs, and when the term chemo or photo is used as the prefix. Okay. So this is the same thing. If you have any doubt in the chart, in this chart, you can ask. Okay. So question number 15. What is the maximum absorption range of bacteriochlorophyll A? So you have to tell the maximum absorption range of this bacteriochlorophyll A. So it is different from the chlorophyll that is present in plants and algae. This bacteriochlorophyll is actually present only in some photosynthetic bacteria, and they have a particular range where they are having the absorption. So what is the wavelength? Option A is between 600 to 725 nanometer. Option B is 625 to 700 nanometer. Option C is 800 to 925 nanometer, and then option D is 825 to 900 nanometer. So you have answered option C. Okay. So the correct answer for this question is option C. Maximum absorption range of bacteriochlorophyll A is between 800 to 925 nanometer. So this, can you tell me in what range does it come? Is it visible light range? Is it UV range, X-ray, infrared? (800 nanometer to 925 nanometer falls in what range of light, of radiation? Any idea? UV, X-rays, infrared. I'm giving you 3 options. Yes, IR beginning. Yes, that's the correct answer. So basically this bacteriochlorophyll can absorb in infrared region. Okay, IR region. So the next question is question number 16th. Maximum absorption of chlorophyll A and B respectively are. Now this is the chlorophyll, these are the chlorophylls that are present in plants, all the plants and different algae. So chlorophyll A and B, maximum absorption of chlorophyll A and B are option A 630 and 430 nanometer and 680 to 415 nanometer. For chlorophyll A and B respectively, we have to answer. Option B is 630 to 480 nanometer and 615 to 480 nanometer. Option C is 680 to 415 nanometer and 680 to 430 nanometer. And option D is 615 to 480 nanometer and 630 to 480 nanometer. So can you answer what would be the range of absorption for chlorophyll A and chlorophyll B? Any idea? So the correct answer is option B, 630 nanometer to 480 nanometer for chlorophyll A and 615 nanometer and 480 nanometer for chlorophyll B. So both the chlorophylls have 2 absorption peak. So I'll show you the spectrum. So here's the spectrum. You can see blue line is for chlorophyll A and red line is for chlorophyll B. So chlorophyll A is absorbing chlorophyll A is absorbing at 480 and 630 nanometers. So you can see at 480 nanometer around there is one sharp peak, and then at 680 nanometer also there is one sharp peak. For chlorophyll B it is 615 and 480 nanometers. So you can see for chlorophyll A and B both they have somewhat overlapping peaks, and then the next peak is at 615 for chlorophyll B and 680 for chlorophyll A. Okay, so this is very easily you can determine the chlorophyll content in any organism if you want, or any plant, any leaf. So if you have a leaf sample, you can simply extract the chlorophyll out of it and see the absorbance. So different chlorophylls have different absorption range, so you can easily quantify the amount of chlorophyll by taking the absorbance in a UV vis spectrophotometer. So this is in the visible range. Basically you can see that visible range starts from 400 to around 700 nanometer, right? From all the rainbow colors that you see, that's in this range, right? So blue being the lowest and red being the highest wavelength. Okay, I hope everything is clear till this point. So moving to the next question, question number 17. Membrane bound pigments that participate in photosynthesis and photoprotection for the organism are called. What is the membrane bound pigments called that participate in the photosynthesis and photoprotection of organisms? So apart from chlorophyll, these organisms have other pigments also. So the options are option A carotenoids, option B carcinogens, option C phycobilins, and option D phytonadione. So the correct answer, you have answered option C, right? Okay, so let's see, the correct answer is option A. So phycobilins are also the pigments that are present, and they do participate in photosynthesis, but then photoprotection of the organisms are carried out by carotenoids. So carotenoids are the class of more than 750 naturally occurring pigments synthesized by the plants, algae, and photosynthetic bacteria. These richly colored molecules are source of the yellow, orange, and red colors of many plants. So you must have seen plants with red leaves. There are some plants and succulents you must have seen with red colored leaves. So that red color comes from the pigment carotenoids. So carotenoids are ubiquitous and essential pigments in photosynthesis. They absorb in the blue-green region of the solar spectrum and transfer the absorbed energy to bacteriochlorophylls or chlorophylls, and so expand the wavelength range of light that is able to drive photosynthesis. So you have seen the chlorophyll A and B. Chlorophyll A and B basically are responsible for the green color. So they absorb mainly in the blue, they absorb the blue light and emit green, right? So and you have seen only the absorption of chlorophyll A and chlorophyll B were there only in the range of 400 something, and then in the range of 600 something, right? So to broaden that range, there are some accessory pigments present in these plants and photosynthetic microorganisms as well. So these accessory pigments, what they do, they absorb at different wavelengths of the visible spectrum. So apart from the 400 and 700 nanometer, we have 500, 600, all those nanometers between in the visible spectrum, right? So these accessory pigments like carotenoids or phycobilins in that case also, they can absorb light at different wavelengths in the visible spectrum, thus increasing the chances of photosynthesis happening at any of the wavelength that is present. So basically it expands the wavelength range of light for the photosynthesis. Along with that, carotenoids have a photoprotective effect. So how does it protects the plant from like how does it protects the plant or any photosynthetic organism? So what happens is carotenoids, since as it is mentioned that they are the source of yellow, orange and red color, so they are transmitting, they are emitting yellow, orange and red light range, right? So they can actually absorb the UV range light, and you know UV radiations are very harmful to the to any organism, as it can cause DNA damage. So you must have heard of cases of skin cancer caused by UV irradiation. So people are worried about the ozone holes because they are causing the UV rays from sunlight to reach the earth's surface directly. And these UV radiations, if they fall on your skin and if you are exposed to UV radiation for a long time, they can cause many mutations in the DNA. So DNA, we have discussed in the previous sessions, one of the sessions we had discussed that DNA is mainly made of nitrogenous bases, right? ATGC, adenine, thymine, cytosine. So those ATGC, those nitrogenous bases, they can get mutated, and because of which your sequence of DNA, so let's say you have a DNA with some sequence that can get changed, and once the sequence of the DNA changes, it can affect the protein that is being translated from that DNA and further affect the processes that are happening in the body, right? So let's say one of my gene gets mutated, and now that gene, whatever protein it was coding for, now that protein will not be functional because of that one mutation, and because of that non-functional protein, that protein is now not doing anything in our body, okay, and thus my body is facing my body has the problem with that UV that I got exposed to. So that is how same goes for plants also. So plants are also living organisms, so they also have genetic material, DNA, okay. Even the photosynthetic bacteria, the algae, every organism has a genetic material, right? Those genetic material can get affected by the UV radiation present in the environment. So carotenoids, what they do, they can absorb the UV rays and they can protect the DNA of the organism from getting exposed to the UV radiation and getting the mutations in the genome, causing harmful effects to the organism. Okay, so that's how carotenoids make carotenoids act as photoprotective agents inside the plants, algae and other photosynthetic bacteria. Okay, so along with assisting in the photosynthesis by expanding the wavelength range, they also have the ability to protect the DNA or the genetic material of the of these organisms. Okay. I hope that's clear. Okay. So moving to the next question, question number 18. Water soluble pigments not attached to membranes are known as dash. Option A, carotenoids. Option B, carcinogens. Option C, phycobilins. And option D, phytonadione. Yes, the correct answer for this question is phycobilins. Option C, phycobilins. So phycobilins are again accessory pigments like carotenoids only, but they are water soluble pigments and they are not attached to the membranes. Okay. So phycobilins, these are light capturing bilins found in cyanobacteria and in the chloroplast of red algae, glaucophytes and cryptomonads. So it is not present in the green algae and plants. So it is basically a different pigment than the chlorophyll. So it is like chlorophyll, but it's mainly so it is responsible for the brownish red color. So it is mainly present in the red algae and some cryptomonads and glaucophytes. So most of their molecules consists of a chromophore which makes them colored. Okay, so red algae gets the red color from the chromophore that is present in the structure of phycobilin. What is chromophore? Do you know what is chromophore? It's basically a structural unit that can absorb a particular wavelength and emit some other wavelength, giving color to the compound. Okay. So all the colored compounds that you can see in your laboratory or anywhere, those have some or the other structure that is giving them that particular color. Okay. So they are unique among photosynthetic pigments in that they are bonded to a certain water soluble protein known as phycobilin proteins. So there's a protein called phycobilin proteins to which these pigments are attached. Okay, and this protein is water soluble protein. Okay, it's not a membrane protein. So phycobilin proteins then pass the light energy to chlorophylls for photosynthesis. Phycobilins are especially efficient at absorbing red, orange, yellow and green light wavelengths that are not well absorbed by chlorophyll A. So you have seen chlorophyll A was absorbing only in the range of blue and the red light, right? So it was absorbing some 400 nanometers and some 600 nanometers. Okay. So other than that, all the other wavelengths of the visible light spectrum, phycobilins actually help to absorb in those wavelength range. So similar to carotenoids, phycobilins also have the ability to assist in photosynthesis by expanding the wavelength range that are not well absorbed by the chlorophyll A. Organisms growing in shallow water tend to contain phycobilins that can capture yellow or red light, while those at greater depths often contain more of the phycobilins that can capture green light. Okay, so at the surface of the water or in the environment, all the organisms have the access to all the wavelength range of visible spectrum, and chlorophyll A and B alone can function very nicely. But organisms that are present inside the water where light is not reaching completely, in those areas very few light is present and mostly wavelengths of red and yellow region, the later region of the visible spectrum is present at these uh, inside these surfaces, water surfaces. So in those region, phycobilins actually help to absorb in those region, yellow red region. Other than that, at greater depth, phycobilins can capture green light as well. So if you go deeper in the water body, you will see the light source will get dim. So the light source will get depleted as you go deeper in a water body, right? So in those cases, when there is no other wavelengths of light available for the organism, how will they do the photosynthesis? They need light energy for the photosynthesis. Okay, so in those cases, phycobilins and such other accessory pigments, they help in capturing the darker colors that are at a lower wavelength, you can say. So they can capture green light, which is relatively abundant at the deeper surfaces. Okay, so if you see a lake or a pond, at the surface of that lake or a pond, abundant light is present for all the wavelengths, but when you go deeper, it becomes darker and darker, and mostly green light is present deep inside, and thus phycobilins can capture those green lights. Phycobilins fluoresce at a particular wavelength and are therefore often used in research as chemical tags, example by binding phycobilin proteins to antibodies in a technique known as immunofluorescence. So this is just an application of phycobilin other than its natural role, since phycobilins have the fluorescence properties. So what do you mean by fluorescence properties? So any substance, if it can absorb one wavelength of light and it can emit at a higher wavelength, so it is called to have it is known to have fluorescence properties. So similarly, phycobilins can fluoresce at a particular wavelength. Okay, when they are irradiated with a particular wavelength of light, they can get excited and emit a higher wavelength of light, and that can be seen as fluorescence. So these phycobilin proteins or phycobilin pigment, it is used as a labeling to tag something. So let's say I have some antibody or some other protein, and that protein does not have any color or fluorescence. So how will I visualize it? So what people do, they tag those protein of interest with these kind of phycobilin proteins or any other. So you have, I don't know if you have heard about green fluorescent protein or red fluorescent protein. All these fluorescent proteins are used for tagging purpose, and after tagging you can track your protein of interest, because now the fluorescence is attached to your protein of interest. So wherever the fluorescence will be there, your protein of interest is also there, and that is called immunofluorescence. So immunofluorescence term is mainly used for antibody based fluorescence. So if you are binding these phycobilin proteins to antibodies, and then you are binding those antibodies to your protein, that is called immunofluorescence. Since you are using antibody, so the term immuno is used. Okay, so that's an application of these colored fluorescent pigments. Other than that, the natural role of phycobilins in the plants I have already explained, to expand the wavelength range of the photosynthetic organisms. Okay, so moving to the next question, question number 19. Number of electrons required for the nitrification of ammonium. Nitrification of ammonium means conversion of ammonia into nitrite and then nitrate. So ammonia is first oxidized into nitrite, and then nitrite is further oxidized into nitrate. So in the total process, what is the number of electrons that are required for the whole process to occur? Can you answer? So I'll give you hint. You can write in your notebooks and try to solve this also. It's not very difficult. So ammonium is basically NH4 plus, okay, and nitrite is NO2 minus. So if you write NH4 plus goes to NO2 minus and try to balance the equation, you will get the number of electrons that are required for this first step. And similarly then from NO2 minus to NO3 minus, you can see the number of electrons required. Do you guys know the answer? Okay, I'll tell. So the correct answer for this question is 8. Yes, Chetna has answered 8. So nitrification, I told you, it's a process in which ammonia or ammonium, like ammonium is the ionic form of ammonia, it is converted into nitrate. So first in the first step of nitrification, oxidation of ammonia into nitrite occurs by nitrosifying or ammonia oxidizing bacteria, and then in the next step nitrites are converted into nitrates by oxidation again by nitrite oxidizing bacteria. Okay, first ammonia oxidizing bacteria are oxidizing the ammonia into nitrite, then nitrite oxidizing bacteria are converting nitrite into nitrate. Okay, so let's see how to calculate the number of electrons. So if you know the formula for ammonium and nitrite and nitrate, it's very easy. So here you can see ammonium ion, that is NH4 plus, goes to nitrite and NO2 minus. So now if you have to balance the equation, so I have 2 oxygen here and 4 hydrogens here. So first we will balance the oxygen. Let's see. So 2 oxygen here, so what we can add? We can add a water molecule. So 1 water molecule has 1 oxygen, so we have to add 2 water molecules. Okay, so we added 2 water molecules on the left hand side. Now if you see, now how many hydrogens do we have? We have 4 hydrogen here and 4 hydrogen here. 2 into 2 is 4 hydrogen here. Okay, 1 molecule of water has 2 hydrogens, so 2 molecules of water has 4 hydrogens. Okay, so we have 8 hydrogens. So we will balance the hydrogen ions then. So we will add 8 protons on the left hand side. Now you can see the charge. What is the charge on this molecule? Plus 1, right? What is the charge on the left hand side? We have plus 8 minus 1, so that is plus 7. So the left hand side has plus 1 charge, the right hand side has plus 7 charge. So if I have to balance, I have to reduce 6 charges from the right hand side to equate it with the left hand side, right? So I can add 6 electrons, since electrons are negatively charged. So 6 electrons means minus 6, this is plus 8, this is minus 1. So 8 minus 1, 7, minus 6 is plus 1, and here also we have plus 1. So our charges are balanced. Now you can see for the oxidation of ammonium into nitrite, 6 electrons are required. Sorry. So 6 electrons are required for conversion of ammonium into nitrite. Okay, so that was the first step. What was the second step? The second step was nitrite into nitrate. So here I've directly written the balanced equation, but you can try balancing it on your own. So if you have NO2 minus goes to NO3 minus, so N is already balanced. Oxygen, you have 2 oxygen on the left hand side and 3 oxygen on the right hand side. So what we will do, we can add 1 oxygen on the left hand side by adding a water molecule, H2O. So one H2O has 1 oxygen molecule. So the oxygens are balanced now. Now once we have added a water molecule, we have 2 hydrogen ions extra on left hand side. So we will add 2 protons on the right hand side to balance that. And now we will balance the charge. So you see on the left hand side there is minus 1 charge. On the right hand side without this 2 electrons, you can see 2 protons are giving plus 2 and then this is minus 1. So 2 minus 1 is 1. So this is plus 1, this is minus 1. So if I have to make plus 1 into minus 1, what will I do? I will add 2 electrons. Okay, so plus 1. So 1 minus 2 will be minus 1, right? So I added 2 electrons. So 6 electrons are required in the first step, that is oxidation of ammonia into nitrite. And then oxidation of nitrite into nitrate requires 2 electrons, right? So total number of electrons are 8. Okay, 6 plus 2 is 8. So if you see in the question, number of electrons required for the nitrification of ammonium. Nitrification of ammonium means ammonia to nitrite, that requires 6 electrons, and then nitrite to nitrate, that requires 2 electrons. So total number of electrons is 8. Option number C, 8 is the correct answer. I hope this is clear. So nitrification is a very important process. You should remember the names of the bacteria that causes all these individual steps. So there are ammonia oxidizing bacteria, there are nitrite oxidizing bacteria. So you should know the names of the bacteria also. Mostly these nitrifying bacteria are present in soil. Okay, so you can see, you can read about it, that in the lecture videos also some of the examples are given. So this is clear. So we'll move to the next question. Question number 20. Order of zones from top to bottom in a Vinogradsky column. So I'll explain what a Vinogradsky column is. Let's see if you know the answer for this for now. Order of zones from top to bottom in a Vinogradsky column. Option A is aerobic. First is aerobic, then anaerobic, then aerobic water, then anaerobic sediment, and then anaerobic water. Option B is aerobic water, anaerobic water, and then anaerobic sediment. Option C is anaerobic sediment, aerobic water, anaerobic water, and option D is anaerobic water, aerobic sediment, and anaerobic sediment. So from top to bottom we have to say. So what would be the first layer in a Vinogradsky column? It would be, any idea what will be the answer for this question? So the first layer in a Vinogradsky column would be aerobic water. Then the next layer would be anaerobic water, and then the anaerobic sediment. From top to bottom, bottommost layer is the anaerobic sediment. Over the sediment you will have the anaerobic water. Over the anaerobic water you will have the aerobic water. Okay, so that's how a Vinogradsky column is arranged. So I'll explain what a Vinogradsky column is. Actually it was developed by Vinogradsky to mimic the conditions that are naturally present. So in a lake or a pond ecosystem, you have the bed of that water body. Okay, so that is deep inside the water body. So there is no oxygen present, because oxygen cannot diffuse to that depth. So there won't be any oxygen present at the end of a river. So that part would be anaerobic, last part. And you know the bed of the river or bed of any water body has sediment. So that is anaerobic sediment. So the last layer, the deepest layer of any water body is anaerobic sediment. Then you have the water. So up to certain point there won't be any oxygen in the water region also. So that would be anaerobic water. And then when you will come even closer to the surface, you will see oxygen will slowly start diffusing. So there will be a region of aerobic water. Okay, so what Vinogradsky did, and you all can do actually, this is a very simple experiment. Basically what you have to do is you take a long tube, a bottle, or a, let's say, measuring cylinder, a long measuring cylinder, and then you can put some soil as the sediment to make the sediment base. Okay, and then you put water over it, so that sediment, and and you can add some nutrients, some carbon source, some nitrogen source, and some sulfur source. Okay, or you can choose a soil that has all these nutrients present in it. So once you add this and then leave the column like that under sunlight, so what will happen, the light will pass through the column till a certain point, and along with light, oxygen will be present in the surface of the column but not deep inside. So the column will automatically get divided into these three zones: aerobic, anaerobic water, and then anaerobic sediment. Okay, and based on these three zones, different kinds of microorganisms will start growing depending on where they can survive. So if it's an aerobic organism, it will be present in the aerobic region only. If an organism can survive without oxygen, like in that chart I have explained, there are certain bacteria, certain organisms that can use anoxygenic molecules as the uh, electron acceptor. So instead of oxygen, they will use sulfur sources. So since you are adding sulfur as the nutrient, so you can find those sulfur bacteria. So like that, all the microorganisms will get divided depending on the zone. So this is a very nice experiment that was designed by Vinogradsky to mimic a natural ecosystem and to understand how different bacteria are metabolizing different components, different nutrients, and one bacteria can actually support the nutrient requirements by another bacteria. So let's see what are the different types of bacteria that are present in these zones. So you have the aerobic zone at the top. It's an open column, so air can easily pass through the column. So in the aerobic zone, you will find some chemoautotrophs. Chemoautotrophs means they are autotrophs, so they can utilize carbon dioxide as the carbon source, but they will use the chemical energy. Okay, so chemoautotrophs, then there will be some cyanobacteria, some oxygenic photosynthesis will be happening in that. So you must have seen in a lake, it's covered with a green algal growth sometimes. So those green algae, mostly they are cyanobacteria. Cyanobacteria are nothing but called blue green algae. So those blue green algae are covered all over the surface of a lake or any water body. That is because they can do oxygenic photosynthesis. They can use oxygen. Basically they can use H2O to reduce the carbon dioxide. Then when you will come further down, there will be sulfur oxidizing bacteria. Those are anoxygenic photosynthetic bacteria. So what they will use H2S as the energy source. So oxygenic photosynthesis, H2O was being used, but by these bacteria H2S, but these bacteria will use H2S as the energy source. Okay, and from where they are getting the H2S, they will get the H2S from these anaerobic sediments. So I'll explain that. So then comes the aerobic chemoheterotrophs. Heterotrophs means they are using organic carbon source, right? Then after chemoheterotrophs, you will have the anaerobic zone. So this was in the aerobic zone. So you had cyanobacteria doing oxygenic photosynthetic synthesis. You had sulfur oxidizing bacteria doing anoxygenic photosynthesis, and then you have chemoheterotrophs, aerobic chemoheterotrophs. They are also using oxygen as the terminal electron acceptor. Okay. Then the anaerobic zone. In the anaerobic zone, you have two parts, anaerobic water and anaerobic sediment. So in the anaerobic water, you will find photoheterotrophs, because light can still reach this part. So there will be photoheterotrophs, that means they can use light as the energy source. So purple non-sulfur bacteria are mainly present in this region, which are using light as the energy source and organic carbon as the carbon source. Then there will be anaerobic photosynthetic purple and green sulfur bacteria, which will use sulfur deposits that are they will use the sulfur as the terminal electron acceptor. They will do anoxygenic photosynthesis. So they are photosynthetic bacteria, that means they are using carbon dioxide as the carbon source. And then at the bottom there will be anaerobic respiration, and there will be sulfur reducing bacteria. So sulfur reducing bacteria, what they will do, they will reduce the sulfate into sulfides. So once the sulfates are reduced into sulfides, these sulfides, since with H2 with combined with hydrogen, it's in the gaseous form, right? H2S is a gas. So once these sulfur reducing bacteria that are present in the anaerobic sediment in the last layer, when they are reducing the sulfates, they are producing H2S, and the H2S, since it's a gas, it can move towards the upper region, and in the upper region you had sulfur oxidizing bacteria, which is using these H2S and converted into like using these H2S as the energy source. Okay. So that's how you can see that some other bacteria is helping for the growth of the bacteria that are present in the aerobic zone. So that's how it works, that's how the nature works, and it's a demonstration of natural ecosystem in a laboratory setup. Okay. So I hope this is clear how a Vinogradsky column is working. Okay, it's a very nice experiment if you ever get the chance to do it. So generally in courses like microbiology, people have this as one of the practicals. So after you set up a column like this and leave it under the sunlight, and then after a week or so you can observe all these layers. So these are visible as different colored layers in the, if you are using a transparent tube, you can actually see the different colors layers, purple, green, like that. Okay, so this is clear. If you have any doubt, you can ask. So moving to the next question, question number 21. Photosynthesis occurs in dash. So option A is chloroplast, option B is Golgi body, option C is endoplasmic reticulum, and option D is nucleus. Where does the photosynthesis occur? Can you answer? This is a very simple question. You must have studied in schools also. Photosynthesis occurs in, yes, option number A, chloroplast. So chloroplasts are basically chloroplasts are basically the organelles inside which chlorophyll pigment is present, and those chlorophyll pigment you have seen, it's the major component for the process of photosynthesis, right? So chloroplasts are the organelles. They are present in all these photosynthetic plants and different microorganisms also, photosynthetic microorganisms, and they are not present in bacteria because, as you know, as I've discussed in the previous sessions also, that bacteria, since they are prokaryotes, they do not have defined organelles. Okay, like eukaryotes have defined membrane bound organelles, but prokaryotes do not have. They just have everything disposed in the cytoplasm of their cell. Okay. So next question is, the final product of Calvin cycle is option A RUBP, ribulose bisphosphate, option B glucose, option C dihydroxyacetone phosphate DHAP, and option D is glyceraldehyde 3-phosphate, that is G3P. So what is the final product of Calvin cycle? So Calvin cycle occurs in all these photosynthetic organisms, and it occurs for the incorporation of carbon dioxide. So I have mentioned all the autotrophs are using carbon dioxide as the carbon source. So Chetna has answered glucose. So actually this is a very confusing part. The correct answer is glyceraldehyde 3-phosphate. So Calvin cycle Calvin cycle occurs as I have mentioned, that autotrophs are using CO2 as the carbon source, right? So that CO2 needs to be incorporated. So you have seen in the previous sessions, aerobic respiration was happening. We studied the different pathways, glycolysis and then TCA, and then it moved to electron transport chain, and at the end the ATP synthesis, right? So in that process, what was happening? Glucose was broken down into carbon dioxide and water, right? Glucose in presence of oxygen, since it was aerobic respiration, in presence of oxygen, it was breaking into carbon dioxide and water. So glucose plus oxygen gives carbon dioxide plus water. In photosynthesis, what is happening? The opposite reaction is happening actually. The carbon dioxide is being fixed. So carbon dioxide and water combines to form glucose and oxygen. But that fixation actually happens through this Calvin cycle. But glucose is not the part of Calvin cycle. Calvin cycle actually ends at the glyceraldehyde 3-phosphate. So here you can see the Calvin cycle. So CO2 is the input source. So CO2 first gets incorporated into CO2 gets incorporated into a 5-carbon molecule called ribulose diphosphate, RUBP. So ribulose diphosphate, since it's a 5-carbon and one carbon is coming from CO2, so it becomes a 6-carbon molecule. That 6-carbon molecule then divides into a 2 3-carbon molecules. So one 6-carbon molecule divides into 2 3-carbon molecules called 3-phosphoglyceric acid. This 3-phosphoglyceric acid then converts into 1,3-diphosphoglyceric acid and then glyceraldehyde 3-phosphate. During this conversion from 3-phosphoglyceric acid to glyceraldehyde 3-phosphate, the ATP and NADPH are utilized, and this glyceraldehyde 3-phosphate is the output of the Calvin cycle. So so what happens is one CO2 can make 2 molecules of glyceraldehyde 3-phosphate, right? Because one CO2 is getting ma'am, when pathway does not stop at glyceraldehyde 3-phosphate, can we say this is the final product? So the pathway, so Calvin cycle is basically the cycling of the whole, so the incorporation of CO2 into RUBP to form the glyceraldehyde 3-phosphate, that is the Calvin cycle. Glyceraldehyde 3-phosphate then leaves the Calvin cycle and 2-glyceraldehyde 3-phosphate actually join to form glucose and other 6-carbon sugars. Okay, so the final product of Calvin cycle will always be glyceraldehyde 3-phosphate. Okay, glucose is not the part of Calvin cycle, although the glyceraldehyde 3-phosphate later leaves the Calvin cycle and then glucose is being formed. Okay, so that is one thing to take care of. So in case of aerobic respiration, or in case of like when we were studying about the about the different pathways, glycolysis and TCA. So you had seen the end product of glycolysis was pyruvate, right? From glucose, 2 molecules of pyruvate were forming, but the starting product of TCA was acetyl coenzyme. So pyruvate, conversion of pyruvate into acetyl coenzyme, that is not the part of glycolysis. Although you can say that pyruvate is further converted into acetyl coenzyme, but pyruvate is also going to other pathways. So the pathway of glycolysis stops at pyruvate. Okay, so similarly in this case, Calvin cycle, the cycle is stopping at glyceraldehyde 3-phosphate. So glyceraldehyde 3-phosphate leaves this cycle. Whatever remaining glyceraldehyde 3-phosphate is there, it will again get recycled to make 5 molecules of ribulose, to make 5 carbon ribulose diphosphate. In this 5 carbon, again a CO2 will be incorporated and become a 6 carbon molecule. That 6 carbon will divide into 2 3 carbon molecules. That 3 carbon molecule will get converted into glyceraldehyde 3-phosphate, which is also a 3 carbon molecule, and similarly the cycle keeps on going. So basically 3 molecules of CO2 are fixed and 1 molecule of glyceraldehyde 3-phosphate is produced and leaves the cycle. 2 molecules of glyceraldehyde 3-phosphate are needed to make 1 molecule of glucose. Sorry, there is a typo here. And therefore the cycle must turn 6 times for each glucose molecules produced. So okay, so this part you should understand very clearly. How many CO2 are required, how many glucose molecules are being made, how many glyceraldehyde 3-phosphates are required to make a glucose molecule, how many ATPs are ATP and NADPH are utilized. These things you have to understand very clearly. And if you see, this is a very nice diagram. This is the diagram taken from Tortora. In your lecture videos also reference is given, so you can download. Ebook is easily available for this book, so you can download this book. This is a very nice representation. You can see this was a 5 carbon sugar, right? Now in this 5 carbon sugar, 1 CO2, 1 carbon is added. It became a 6 carbon sugar. Now 1 6 carbon sugar can divide into 2 3 carbon sugars. So now if 3 CO2s are entering, that means 3 RUBP plus 3 CO2 will give 3 6 carbon sugar, and in turn 6 3 carbon sugars. Okay, 6 molecules of a 3 carbon sugar. This 3 carbon sugar will be converted into 6 molecules of glyceraldehyde 3-phosphate, and in this process 6 ATP and 6 NADPH will be used, okay, to make 6 molecules of glyceraldehyde 3-phosphate, and from this 1 molecule of glyceraldehyde 3-phosphate is leaving the cycle, and 5 molecules are recycled again to make the RUBP, and there also 3 molecules of ATP are being used. So you can see 9 molecules of ATP and 6 molecules of NADPH is used in one cycle, and then similar, and since 2 molecules of glyceraldehyde 3-phosphate are required for 1 glucose molecule, so one more turn of this cycle, so that means 6 plus 3 9 into 2 18, so 18 ATPs and 6 into 2 is 12 NADPH molecules are required. Okay, so 6 molecules of CO2, 18 molecules of ATP and 12 molecules of NADPH will give 1 molecule of glucose. Okay, this cycle is very important to understand the photosynthesis process. This cycle and all the photosystems that are discussed in the lecture videos. I would suggest you to go through those things clearly. If you have any doubt, you can ask me in this session or in the next session also. So just a second. So moving to the next question, question number 23. Reduction of sulfates and sulfides to hydrogen sulfide is done by which group of bacteria? So we had discussed the Vinogradsky column right now, so you should be able to answer. Which group of bacteria was converting sulfates and sulfides into hydrogen sulfide? Which, or tell me the region only, that will be fine. So option A is aerobic sulfate reducing bacteria, option D is photosynthetic sulfur bacteria, option C is anaerobic sulfate reducing bacteria, and option D is heterotrophic bacteria. So you had seen in the Vinogradsky column, H2S was being released upwards, right? So yes, the answer is option number C, anaerobic sulfate reducing bacteria. You are right. So you had seen in the Vinogradsky column, the sulfate and sulfite, basically it's the sulfate reducing bacteria were there at the end of the column, and they were reducing the sulfates into sulfides, ultimately to sulfides, and that is combining with the hydrogen to make hydrogen sulfide, and hydrogen sulfide being gaseous gets released to the upper surface of the column, where the N-oxygenic photosynthetic bacteria can use H2S as the energy source. Okay. So this was the last question of the session. So if you have any doubt, you can ask. I would suggest you to understand the Calvin cycle, the photosynthetic pathways, how the photosystems work, how the phosphorylation is happening. So since you had seen the phosphorylation process of respiration, so how ATP was generated in most of the animals and other microorganisms. In case of photosynthetic bacteria and in case of plants, the process is slightly different. So you can look into it, and then you can let me know if you have any doubt. I'll try to clarify it in this session or the next session. So today is the second last session, so we'll have only one more session in the on next Saturday. So then I think you guys have your exams for this course. So if you have any doubt, you can ask. Do you want me to project any other question from the session? Again, I can do that. Or if everything is clear, then we can end this session. Okay, so we'll end this session now if you don't have any doubt. Thank you so much for attending. We'll see you next Saturday. Thank you.